The benefit of filtering blue light with yellow-tinted intraocular lens is a raging debate. Proponents of blue light filtering talk about many advantages of this filtration, while some dispute the benefit of blue light filtering IOLs. In this video, we will look into the advantages and the proposed disadvantages of blue light filtering IOLs after cataract surgery. First, what is blue light? Blue light, part of the visible light spectrum, is emitted from the sun, which is the most powerful source of blue light, as well as artificial sources, including digital devices. While even indoors, the sun is the largest source of blue light. Blue light is also found in the LEDs of our domestic lighting and our screens, though the illumination levels are lower. The visible spectrum of light is from 380 nanometers to 700 nanometers. Out of this, short wavelength high-frequency light is considered from 380 nanometers to 500 nanometers. This is the zone of blue light. Blue light filtering IOLs often filter the shorter wavelength blue light, usually below 450 nanometers, and allow more amount of longer wavelength blue light that is above 450 nanometers. Why is shorter wavelength blue light harmful? Photoreceptor cells like rods and cones are specialized cells in the retina that convert light into signals that the brain can use for vision. Photoreceptor cells convert light into neural signals that are sent to the brain for image processing. They are responsible for color vision and night vision. Beneath the photoreceptor cells is a unique underlying cell called the retinal pigment epithelium. The retinal pigment epithelium, or the RPE, provides important nutrients and nourishment to the photoreceptor cells. The photoreceptor cells metabolize and hence produce or excrete waste materials called lipofuscin. The lipofuscin is flushed out of the retina through a process called phagocytosis by the RPE cells. However, as we age, the process of phagocytosis slows down and lipofuscin starts accumulating. In the lipofuscin, there is a particular molecule called A2E that reacts with shorter wavelength blue light. This often leads to atrophy of the cells, mainly the RPE. Proponents of blue light filtering IOLs often connect the death or atrophy of such cells to age-related macular degeneration. The other side. On the other hand, Martin Mainster, in an article published in 2009, refutes this claim. He writes that acute retinal phototoxicity experiments on cell cultures and experimental animals have been used to advocate blue-blocking IOLs, but AMD is a chronic process, whereas photic retinopathy occurs only when brilliant light exposure acutely overwhelms retinal defenses. Acute phototoxicity can injure the retina, but it cannot simulate AMD, just as sun gazing can scar the fovea, but it cannot simulate a lifetime of normal environmental light exposure. Are there any other potential benefits in filtering out short wavelength blue light? While there are conflicting schools of thought on the role of blue light filtration vis-à-vis intraocular lenses, proponents of blue light filtration may look towards its role in improving macular pigment optical density, or MPOD. To this school of thought, one of the most important benefits of filtering short wavelength blue light is to protect the macular pigment and thereby increase MPOD. Unfortunately, though the macular pigment plays a crucial role in protecting human vision, this is one of the least talked about subjects. Interestingly, the importance of macular pigment was not a recent discovery, but dates back to the works of Max Schultz in the 19th century. He theorized that the yellow spot absorbed blue light. Many decades later, in the 1940s, G. Wald hypothesized that the macular pigment consisted of specific carotenoids. Later, in the 1980s, Bone et al., Ham et al., and various other studies concluded that the macular pigment consists of lutein and zeaxanthin, and has a definite role to play in human vision. What is macular pigment? Human macular pigment consists of lutein and zeaxanthin. They reach their greatest concentration in the fovea and diminish as you move further away from the fovea to the peripheral retina. However, though the carotenoids are concentrated in this region, lutein and zeaxanthin are found in the whole of the retina. What specific benefits are attributed to macular pigment? Extensive research, starting with G. Wald to modern-day studies, has indicated a multifaceted beneficial role of the macular pigment. The absorbance power of macular pigment of the blue light peaks at 460 nanometers of wavelength and therefore reduces the sensitivity of the macular region to short wavelengths of light by acting as a pre-retinal filter. The fovea has the greatest visual acuity of all retinal regions because of its closely spaced cone receptor cells. This, however, makes the fovea vulnerable to any form of image degradation that may result from higher-order aberrations, one of which is chromatic aberration. In 1866, Schulze first indicated that the image-degrading effect of chromatic aberration might be mitigated by the filtering properties of macular pigment by preventing short-wavelength blue light from reaching the cone cells. This was later corroborated by G. Wald. In short, macular pigment helps the eye to minimize the effect of chromatic aberration, thus reducing its effect on human vision. Other benefits include the macular pigment's antioxidant properties. The generation of reactive oxygen species can occur as a byproduct of cellular metabolism or as the result of photochemical reactions. These are free radicals, which are partially reduced oxygen species containing one or more unpaired electrons. These molecules are highly reactive and can result in cell death. The retina may be particularly susceptible to damage by free radicals. The macular pigment may act to protect the retina by acting as a free radical scavenger and reducing the effect of photochemical damage. Glare is a common phenomenon among many elderly patients suffering from retinal problems. Studies like the Beaver Dam have indicated a close relation between lower macular optical density levels and glare phenomenon. What is the role of blue light filtration and macular pigment? If macular pigment is so important for healthy vision, then it may be important to protect the macular pigment and help maintain a healthy macular pigment optical density, or MPOD. In a study by John M. Nolan, published in the Investigative Ophthalmology Science Journal in 2009, the role of blue light filtering intraocular lenses in augmenting MPOD levels was proved. The MPOD levels were measured by heterochromatic flicker photometry. The basic science behind this test was to ask the patient to observe a light stimulus that was absorbed by the macular pigment and another band of light that was not. The patients were asked to adjust the radiance of the wavelength band absorbed by macular pigment to minimize their perception of flicker. This gives an idea of the macular pigment of the patient. Several commercial devices are also now available that measure macular pigment optical density levels. The proponents of blue light filtering intraocular lenses often link the benefits of blue light filtration to retinal health in the form of arresting programmed cell death of the retinal pigment epithelium. While that line of argument is often talked about, less light is shed on the connection between blue light filtration and healthy levels of macular pigment optical density post-cataract surgery. For more information, follow quickguide.org.